It's wonderful to be out here and to share some of my work. I'm not going to focus too much on slides and graphs and charts and stuff like that, but I do have some very interesting stuff to show you from deep-sea trenches, including some new species that we discovered that are a surprise to us every time that we go. And I hope you'll enjoy this, but also just get a better understanding of the very deepest parts of the ocean. So the average ocean depth is 2.5 miles, and that includes even Hanauma Bay out here. If you average Hanauma Bay's depth in, which is only a few hundred feet at the deepest maybe, to the whole ocean, it's 2.5 miles deep. The deep-sea trenches, which we're going to be talking about today, they have an average depth of 5 1/8 miles, and at the very deepest point, they're about 7.5 miles deep. These trenches, while very deep, they account for less than 0.25% of the ocean. So they occupy a large area just because of their depth and size, but they're a very small part of it. And today I want to show you one reason why we need to consider these more, even though they're a very small part of the ocean. We consider hadal ecology, or the ecology of trenches, any area in the deep sea that lies below 6,000 meters. And so that means, and we seek to describe the abundance, the diversity, and the physiology of animals that live at these depths. And physiology is just how things breathe, how their cellular function works. Basically, everything that we do every day, from breathing to eating to many other parts of our life. And so by knowing that, we seek to understand why they live at these depths and why certain animals start to disappear. We know that fish can only live at about 8,000 meters. We're still trying to find one that's a little bit deeper, and I'll show you some that get close here in this video. Sharks don't go much below 3,000 meters, but invertebrates go all the way to the very bottom. And some of those invertebrates, especially the supergiant amphipods, we'll show you today. So hadal ocean depths, if you look at this schematic, you'll see that the ocean is divided into different provinces. The littoral zone is what we see outside our window in Hanauma Bay. Below that, we start getting into the sublittoral zone, which means things below the water line, and the bathyal and the abyssal zone. And these are basically, the bathyal and the abyssal zones are what we consider the deep sea, anything below 500 meters or so. And you see a variety of vehicles here, Alvin probably being the most famous, but these are other ones. The Alvin, of course, and Jason are the ones that first profiled the Titanic, so that's probably why they're most famous. And these can dive to 6,500 meters or so. But when we get below into the trenches, you'll see the 6,000 meter line. There's very few things that have been down there. Unfortunately, 2 of these 3 vehicles have all been destroyed by hadal pressures. Luckily, when you go this deep, usually there's no people in it, so it is a loss of equipment, but no manpower. The Challenger Deep in the Mariana Trench is the deepest spot in the ocean. It's just at 11,000 meters, which is 7.5 miles. And at that depth, organisms and vehicles experience 1,100 times the atmospheric pressure that we get here today at sea level. And that's about 16,000 pounds per square inch. So if you can imagine having a large dump truck, or maybe a garbage truck balanced on your big toe, that's the type of pressure that creates. Pressure increases with every atmosphere. That's 10 meters. So if you dive down to 10 meters or 30 feet out in the bay, you'll experience twice the pressure that you would at the surface. And it keeps on going like that. And in the deep sea, what we know is that temperature and food supply decrease as depth increases. And basically the reason is that you're getting away from the sunlight. And even in the deepest parts of the ocean, most of your animal life is dependent on things that are produced at the very surface. And these are tiny plants. They're eaten by smaller zooplankton, or small animals. And these sink to the bottom of the seafloor when they're dead. And this provides most of the food that animals need. Now, there are a few exceptions, like hydrothermal vents and other areas where food is produced in the deep sea. But to a majority, everything depends on the things that fall down from the surface. And the further you get away from the surface, the less that food becomes. Except in deep-sea trenches. And actually, it gets to the point that there's so much pressure above these trench areas that the water is compressed enough that you actually get heating. It's a very small scale, maybe 0.5 degree or 1 degree Celsius, but at the bottom of these trenches, the water is actually warmer than it would be at the abyssal depths of 6,000 meters. And the other reason that this changes in trenches is they act as a funneling system. So this rain of organic debris that fuels ocean life on the deep sea floor is actually concentrated because of the steep topography, and you get things like underwater landslides that move more of this food down into the bottom of the trench axis. So these are very different places than most of the deep sea. The temperature doesn't make a huge difference because it's a very small increase, but the food is. At least that's what we think. Most trench research has been very sporadic because it's very hard to get to. So a lot of trenches have either been sampled at the very deepest point, because that's always an interesting place to go, just so you can say that you've sent something there, or the very shallowest points, because maybe that's as far as you can go. And in fact, one of the major problems is most oceanographic research is done by taking samples on the end of wire. And unfortunately, once you get out about 6,000 meters of wire, it becomes so heavy, it would actually rip itself off the ship at that depth because of just that amount of wire that's being spooled out. And if you want to get even further, then you need more than that. So the only ship that's ever done that is a specially designed Japanese ship, and they basically designed a whole research vessel around a trawl winch that could carry those weights. So there are things that are challenging about hadal research, just like anything that you can imagine, especially going into outer space. We don't have quite the budget of NASA, so we do try and make do with what we have. Trenches, for the most part, are formed where you have one piece of an oceanic plate subducting underneath another. Patty Fryer probably talked about this last week, and in fact, this is one of her illustrations. And so this is part of the oceanic plate, and it's a little bit denser. It means it's going to subduct underneath another plate, and that pushes up big islands like the Mariana Islands and Guam. But what it does is actually drags this piece of the ocean down along with this one and creates these super deep depths. So these are not troughs, they're not deep spots, they're actually something that's caused by huge geological pressure and temperature. And of course, what's actually happening here is this old oceanic crust is being dragged down into the mantle where it will be eventually recycled. And that's why in the Pacific, where most of these trenches occur, we have called the Ring of Fire. Because this intense geological pressure brings crust down, but then it brings it back up in the form of volcanoes. And then, and of course, the slippage along these crustal boundaries creates lots of different things. So that's a little bit about trenches and why they're formed and some of the unique biological and physical processes there. But what we want to understand when we started out on this project, which is part of a large international group of researchers from all over the world, we wanted to understand how the combined effects of extreme pressure and ostensibly this increasing food supply impact the ecology of the animals that live there. So part of that is doing a survey of animal densities, and this includes getting sediments from the very deep sea floor and the water to study bacteria. Another large portion of these ecosystems are the sediment invertebrates. So these are things like worms and small crustaceans, and even mollusks, which are bivalves, and snails. And then the other part is we want to know how the scavengers and predators react, because these are the largest animals in the deep sea, and to some degree they're the easiest thing to see. In biology, we call this charismatic megafauna, things like whales and turtles, because they're so interesting to look at. Otherwise, you need lots of microscopes and imagination to imagine some of these things. What we also want to do, and this is part of my research, is to measure the community and individual energetics. So when you took a deep breath earlier, you obviously realize that you do that, and even deep sea cucumbers or snails or anemones, they all breathe as well. But the other thing that happens is that sediments breathe. And it's not so much the mud, it's all the bacteria and animals living in that. And so those are the two things that I've been able to measure in these deep sea trenches, are the measurements of oxygen consumption of individual animals, as well as total community respiration of the sediments. And one of the ways that we do a lot of these surveys is that we do have some instruments that allow us to take samples, but we also do surveys of animals attracted to bait. And this is one thing where we see a lot of these scavengers and predators. And hopefully you'll get to see some of that in the videos that we have in these deep sea areas. So the first research expedition that we took was to the Kermadec Trench, which is this large trench right here above New Zealand. You see in the south western Pacific. This is the Tongan Trench up here. There's a very small, shallow sill depth here that keeps these two from being in the same trench. And here's a close-up view. And this is where the Pacific plate is subducting under this Indian plate, and it creates this large, deep rip up from New Zealand. And we did this last summer, or sorry, last April and May, on a 40-day cruise aboard the RV Thompson, which is from the University of Washington. And we deployed baited traps. We deployed automatic landers that went to the bottom and came back. And most importantly, our main tool was a particular vehicle that I'll show you in a little bit here that is the only one capable of going to these depths. So this is a baited camera. And these things by Alan Jameson and Tom Lindley at the University of Aberdeen in Scotland had made over 200 dives to these ocean depths in the Kermadec Trench. Now what you're seeing here are snailfish. These are some of the world's deepest living fish. And they're not eating the bait per se, which is this large chunk of mackerel. What they're doing is they're sucking off tiny crustaceans, tiny amphipods that are attracted to the bait. And so I think the first thing that you would think is that maybe things that live very deep at much pressure are very sluggish. But what you can see here is these snailfish are very active. And this is not sped up or anything like that. And so these guys, despite the limitations of pressure and cold, they're very active. And if the video will start over here again. The other thing that we learned is these supergiant amphipods. Now you see some tiny amphipods and shrimp here. And in a second, you're going to see a large one coming. And this thing is about the size of a dinner plate. Now, normally everywhere else in the ocean, they're about the size of a quarter, and maybe some other ones get to be the size of a teacup. But what we learned is that even though this guy is huge and we thought he was a dominant predator, you see him just swim right into the ocean. So they're not as smart, and they don't necessarily have all the sensory, abilities that we would have expected. So that's one of the first things that we've learned, because for the most part, these things have been captured in traps. And we can't see how it behaves when it returns to a trap. But as you can see, he kind of gets scared away by the fish milling around, and he does dive right into the ocean. So part of the research is still new to us because nobody has seen this before. And even though they have done over 200 videos of this, the behavior of these large giant amphipods is quite rare.
Do you think that they're that big down there because of lack of predators?
We know that that's part of it. Because once you get past about 8,000 meters, there's no more fish that predate on them. We also know that they have specific adaptations, like a big thorny tail, so that when they do attach to a piece of bait, any animal that tries to eat them is just going to get a mouthful of these quills. So it's almost like having part of a porcupine on your tail. As I mentioned earlier, the reason that we were able to do this research in the Kermadec Trench is this unique vehicle. The hybrid, remotely operated vehicle Nereus. This is designed at Woods Hole. It's very different from most other ROVs because it uses a fiber optic cable for communication. And this helped combat the earlier problem that I mentioned of having too much cable out in the water. It would sink this thing effectively. You can go to the full ocean depth of 11,000 meters. You can't see it here because it's not available, but there's a small fiber optic cable that goes up to a larger depressor weight that communicates to the ship. It's used hundreds of ceramic spheres, which you see contained in here, and then these floats are also full of them. They're about the size of a softball, but by making them that small, they're able to withstand these intense pressures. And it uses different things that we can sample things. It's got a manipulator arm right here so that we can grab things. It also has a suction sampler, which you can't see in there, but it's like a large vacuum tube, so you can vacuum up animals on the ocean floor. And now this thing is also unique because you can take off this lower portion and program it remotely to do transects, or basically what we call mowing the lawn, and using its sonar or its cameras to take pictures and work autonomously without any input from the surface. But for the most part, our work was done with this on, and that way we have a pilot on the surface that communicates with it and controls its actions on the seafloor. Of course, I mentioned earlier that this thing is no longer around because on our very deepest dive in the Kermadec Trench at 10,000 meters, the screen went blank, and half a day later, we started finding debris floating on the surface. So it did make it to quite a few things, and it was in operation for 2 or 3 years, but in the deep sea, especially in trenches, you do have the unfortunate occurrence of things breaking on you. But for its time, it was quite remarkable, and we're hoping that they're going to go. My particular work was this in situ megafauna respirometer, which just means it was able to incubate animals on the seafloor. And if you've done some experiments in school, you can incubate cockroaches or baby chickens or anything like that. And basically, it just means to put them in their own container and measure how much oxygen they use. And from that, you can understand how fast their metabolism works. We built this here at the University of Hawaii, and this is before it's getting a test in shallow water. These small chambers have a flip lid on it, and Nereus was able to suck up animals and put them in there and close the lid. And then it took it to an elevator that allowed us to transport this whole thing back and forth from the surface to the deep sea floor. And it encompasses this titanium housing that holds our electronics. This is an oil-filled volume that allows us to connect all of our electronics and sensory things. And then there's a battery back here that can go to the bottom of the ocean. And on top of this, we would put this top hat. And each one of these is a pump and an oxygen sensor. And that way each chamber could be circulated and the oxygen change could be measured. And so why we wanted to know this is we wanted to know, are these things breathing faster? Are they breathing slower? Are there something that's limiting them? And so I'll show you briefly some of this the results of this work from the Kermadec Trench. And so you've seen these same guys before. And part that we were excited about this is because every once in a while we're down there, we're completely surprised. This happened in the Kermadec Trench as well as the Mariana Trench. And we knew we were going to find snailfish, and we knew we were going to find giant amphipods, and even things like this, which is a crinoid, which is another type of echinoderm, and it sits there and filters things out. And we also see some fairly large shrimp swimming around back here. So obviously the behavior of these animals is something that's new to us. But while we were looking for animals to put into this, there was a few things that showed up that we didn't realize. And that's this guy. Can anyone guess what this might be?
Underwater centipede.
Very close, very close. And in fact, when we first saw it, that's what we thought it was closely related to. It was an isopod. But we didn't know for a long time, and we dissected this video apart for quite a while, trying to figure out what this was. Finally, we captured one. We put it in the chambers, and we were able to do some experiments on it. Even then, we didn't know what it was until it got back to the surface. And in fact, it's a worm, a very large worm, a polychaete worm. And they live in reefs. They live in mud flats. They're just all over the ocean floor. But this guy is incredibly large, almost 5 centimeters long and weighing 2 or 3 grams, which is giant for us. So that's why we had no idea it was a worm. But we do happen to have some very good respiration measurements from it. And in fact, on the very last dive, we collected 2 more of these guys. They never came back because the Nereus imploded. Unfortunately, but this is something that's completely new to us. And I think that's one of the reasons I like deep sea biology so much is there's still this sense of exploration and discovery that occurs, not only at the deepest depths, but even in shallow water. So the results of this, and you can see what this worm looks like right here. It's got some long sensory palps. These are little spines on the sides that he was crawling along with. So it's a very unusual behavior. And you can see right here a sea cucumber inside one of the chambers when we bring it back up. This is another type of sea cucumber called a sea pig. And sometimes these guys rove across the bottom in large herds, grazing the sediments as they go. And this other guy is a sea spider. And he snuck into one of these chambers attached to an anemone. So what we found out is that the holothurians, the sea cucumbers, and the anemones are very similar to rates of other animals. Of course, we correct this for their mass and for temperature. Otherwise, we wouldn't be able to make the right accurate comparison. And what we found out is if you counted all the sea cucumbers for for the area down there, and you multiplied that by how fast they breathe, and did some conversions to carbon, we found out that they respire 0.02 to 0.5 milligrams of carbon a meter squared per year. So numbers don't mean anything without context. So let me just tell you what we believe this means. And we think that this means that if you counted up all the animals down there, there's actually a whole lot of carbon being turned over. And that's just a way for us to measure how much food is down there. And so this is very similar to something that you'd find at much shallower depths, say maybe 1,000, 2,000 meters. So this helps us, I guess, kind of get to the point where we're starting to be able to show that, yes, food does increase in these deep-sea trenches. And why is that important in today's world? Well, one of the biggest things we hear about is carbon sequestration. Whether it's about the carbon that's being released into the atmosphere in the form of carbon dioxide from coal-burning power plants, cars, cement factories, and how that impacts overall global climate change. But what we would hope is that in the deep sea, some of this carbon is being sequestered, basically locked away so that it's not going to get back to the surface ocean where it might be released into the atmosphere. So if it's tied up in these animals, not only in their body mass, but also as they incorporate it through respiration, then we can start getting a better idea of how much carbon is, I guess, sequestered in these animals. And that's important for the overall carbon budgets of the globe. So we're still adding little bits and pieces to this, and hopefully some of this research will allow us to make better estimates of this. So like I said, I won't show you too many graphs, but here's a couple. And so from the Kermadec Trench, we put holothurians, and anthozoans or anemones. Ophiuroids are small brittle stars, and the polychaete worm. And basically, if we just look at the sea cucumbers, we see here at 4,000 meters that they are actually breathing less than these ones at 7,000 and 8,000 meters. So this is exciting to us. This again shows that there might actually be more carbon and more food in the deep trenches than we thought. Of course, you have to look at this with a grain of salt, and we do this by normalizing that for body mass and looking at this off of a larger portion of other studies. And what we see is that actually this line is fairly straight. So when we correct for body mass and temperature, they're actually not breathing that much faster or slower than any other animal based on their size. And one of the general principles of physiology is that a mouse breathes a lot faster than an elephant. And it breathes so much faster that even though its body size is very small, the total amount of oxygen it respires is larger than an elephant. So this is something that we'd expected based on the theory of metabolic ecology. So we're not too surprised by this, but again, it's just good to know this because without actually doing these experiments on the seafloor, we could guess a lot, but we would never know. So hopefully, like I said, we'll be able to do this some more in the future, but it is going to take the new ROV for us to do this in the trenches. Okay, so that's the Kermadec Trench, and I think we saw a few interesting things there. We saw this giant polychaete that we had no idea was around. And so it's always fun on the research vessel late at night when we've collected all of our equipment, we're downloading data and looking at videos to sit around and see something that we never expected. Even for some cynical scientists or people that may have seen it all in the deep sea, there's always something new that surprises us. And so that's why we were a little worried about going to the Mariana Trench because this happened last year in November and December. We didn't have the HROV Nereus anymore, so we had to come up with some new ways to perform these experiments and continue our research. And now we're going to the Mariana Trench, which is the deepest place in the world. It gets very deep in terms of the total trench, but it's also a lot closer to land. So it has some differences from the Kermadec Trench. And so this is the view of the Eastern Pacific. You can see the Philippine Islands here, and Japan is up here. And this is a long line of trenches that are divided up into the Mariana, the Izubonin, the Japanese Trench, and then up here is the Aleutian Trench. If you look at it a little bit closer, we left out Guam right here. And so Guam's a great spot to go to the Mariana Trench. I don't think you new people will go to Guam otherwise if they weren't going there, but it's only about 60 miles from the trench. So it takes about 4 hours to get there by research vessel. As you can see, just from this topographic relief in Google Earth, although they don't send a Google car down there, they do have some very good maps. You can just see how deep and steep this trench is. So these amphipods that we see in the deep sea, they clean up everything. And that's why they're great to study. This is a 4-hour time-lapse camera. That's a large piece of mackerel that weighs about 6 pounds. As you see, you have a light in there. And in a little bit, there's no more bait. That only took about 3 hours for these guys to clean up. They're not very big. Sometimes we'll empty our amphipod traps and they'll just be full. You have to shake them out to get them out. But it's a wonderful way to collect animals, but it's also a little bit scary to think about. This is what happens to things that die and sink down to the bottom of the ocean. So it makes you rethink being buried at sea sometimes. But you know, whether it's earthworms or amphipods, different things can happen. So again, we're always trying new things. This was an extra camera we had, and we put it in there, and it was a little creepy to find out this is what happens. But we knew that in the first place. So like I said, we lost the Nereus, and so I had to come up with a new way that we were going to be able to measure some type of physiological experiments. And so with the help of some engineers at Woods Hole and some money from Schmidt Ocean Institute, we came up with this, the free vehicle coring respirometer. And so this is another picture of a good deep sea elevator. Oops, sorry. It's got a flotation package up here. This is syntactic foam that won't crack or break at pressure. It's got a beacon and a light up here that allows us to find it on the surface. And then down below, there'll be a weight suspended here. And this whole thing will land on the seafloor. And when we're ready to recall it, we'll send it an acoustic signal. It'll drop the weight, which will make it positively buoyant, and the whole thing will float back up to the surface. Now, what we did to increase the amount of samples that we could get is we wanted not only to be able to measure respiration in sediments on the deep sea floor, we wanted to get some of that mud too. Because without a vehicle to get down there, we were going to be lacking on any type of samples that we were going to get. And so the way that this worked is we used an old a spare thruster motor from the Nereus, since it wasn't going to use it anymore. And that turned a large drive screw that went down the center of this. And basically what it did is it pushed these cores into the mud after being on the seafloor for a certain amount of time. We again used another battery, the ubiquitous, these big orange deep sea power and light batteries. And what we did this time is we had 4 different cores on each side of this, and they're just maybe 10 centimeters across, about 4 or 5 inches. And again, on the tops, we had an oxygen sensor and a pump that would circulate water and measure oxygen. And that way we could determine how much oxygen is consumed by sediments over time. And then at the very bottom, they have a very clever device. It's actually just a big swing arm with a piece of carpet on it that will catch the cores when they pull back out of the sediments. And so that way, when it comes back up, we will have not only measurements of the oxygen consumption, but we should have some money. And I'd like to say that it worked pretty well. And here is our one very successful, where we got 4 complete cores. You can see the mud's in here. You can see there's nice clear top water on top. And this is what it looks like, at least from one side of the trench. And what we found out is that on the one side of the trench, on the upriding plate, which is closest to Guam, there's very deep, dark sediments. And to us, that means that one, not only is it organic rich, but it's also being deposited in fairly regular intervals. On the other side of the trench, the downriding plate, we saw that they're strongly laminated, or basically there's lots of different levels to it. And these lighter gray patches are ash or volcanic eruptions. And so we know that these are accumulated over quite a few years. In fact, sometimes tens of thousands of years to accumulate that much mud. What we did discover is that carbon turnover, based on our oxygen respiration rates, are similar to much shallower depths. In fact, a lot of our rates were similar to something that would have occurred in 1,000 meters. Not 6,000, not 7,000, not 8,000, where we did these experiments. And so that helps us to understand that both sides of the trench are actually depot centers. And by that, we just mean they're areas where lots of this falling food and organic matter accumulates on each side of the trench. We also found out that it's not always like that. The very first deployment, we landed it right on a big rock. So when the motor pushed it down, it basically just lifted the whole thing up off the seafloor. So there is a lot of trial and error, but we enjoy it regardless of that. So I'll show you some quick results from this. These blue bars are the amount of oxygen consumed by the sediments in the trenches. Along the bottom, you have an increasing depth. From our experiments, we're at 6,800, 7,900 meters deep, and you'll see that these values are much similar to something that we'd see in 1,000 meters. So on the deep sea floor, just adjacent to us, there's very little oxygen consumption going on, which we interpret to mean there's actually very little carbon, very little food reaching the seafloor. There are anomalies. This large block is underneath the California current upwelling. So in upwellings, you get lots of biological productivity and you get lots of deposition to the seafloor. So this is great. This is exactly what we wanted to see to show that these processes are occurring in much faster rates, which to us means that there's a lot of food reaching the seafloor in these trenches. When you look at it as a proportion of the surface carbon production, which is done by plants, you'll see in red, and the proportion of seafloor carbon turnover in the blue, what you'll notice is that everywhere else, regardless of the amount of carbon overturned, the amount produced on the surface is it's much, much more than that done in the rest of the ocean, except in trenches. This is the mean of our results. You see that the blue here is many times more than what's turned over on the surface. So to us, this is a very, very good example of how, even though there's very little production in the surface, it's all being funneled into these trenches. And so that this is a very good indicator of why there might be so many fish and invertebrates living in these trenches, and it's because this funneling effect is providing lots of food. So this is a video of FBC on the floor, and this was by far the most exciting moment that we had on this cruise. This fish, never seen before, swam into the video one night. We had this video camera on there just so that we can monitor the experiments. And I'm just going to let you watch it because it is something difficult. And it looks like it might be made out of tissue paper. And we've had many ideas of what to call it.
Is it blind, you think?
We think that everything down there is blind. There's so little light that there's no reason for their eyes to work very well. And so even in this light, it's incredibly bright down there. It doesn't seem to have much impact. So blind may not be the right term, but their eyes are so sensitive to light that anything like this will basically affect them. So here he comes again. We sped up the video and clipped out all this stuff. This is the deepest living fish that's ever been documented, down to about 8,145 meters. We see other snailfish, and in fact, we believe now that there's a single species of snailfish in each trench. But they've been living in these trenches at these depths long enough for there to be a different species in each trench. So we call them angelic snailfish, or an ethereal snailfish, or a ghostly snailfish. Hopefully, one of these days we'll actually be able to track. That's the one problem. We have great videos of them, but until you have them in hand, it's very hard to describe new species. So this is one of those serendipitous events that occurs when you're doing science like this, when you never know what to expect. Something amazing will just swim out of the darkness and right into the middle of your experiment, and you completely forget about what's happening in the body and how much oxygen is being used or how well your piece of equipment is performing or not. And you see something like this. Yes, go ahead.
Speaking about these miraculous discoveries in new fish species and all, I asked last week because I read recently, and I'm just told to wait and ask you.
Okay.
I read about this new fish that was found at fairly deep levels. I don't know the exact area, but it's warm-blooded. And she said, you would know all about it and tell us about it.
Well, these fish are warm-blooded anyway. What we've been learning is that, I think that we have lots of these preconceived notions about how they adapt to these environments. We thought, well, it's so cold out, they have to have some. But I think the fact that this new fish, it's not a new fish, we've known about this fish for a while, but new research has revealed that it actually does perform more like a warm-blooded animal than a cold-blooded reptile. And to some degree, that may just be an adaptation to handle the amount of food that it's getting. But it could just be a bizarre physiological phenomenon that makes it different than many other fish. So I think that will spur more research in terms of trying to figure out if this is a common adaptation or if this is a rare.
So you're saying basically it might be because of the depth at which it lives, and it might be much more generally true for other species than we know because nobody's done the research yet?
Exactly. Some of the researchers that were with us, we were very interested in determining how much particular protein stabilizers there are in this fish. And so the snailfish are the deepest living fish in the ocean. We have videos of many other fish, but they don't get much beyond 6,500 meters. And that's because the snailfish have a large amount of TMAO, which is short for large chemical, and it stabilizes proteins. At depths, physiological or cellular activities are hard for things to occur because the pressure actually deforms these proteins. And TMAO, which is actually the chemical that makes fish smelly, or makes them smell like fish, it protects these proteins. And these snailfish have a much higher proportion of those. And so I think if more research is done into the actual chemistry of this fish, the biochemistry of its reactions, we may find out that there's TMAO and maybe some other chemical that allows it to do that. And that, in concert with this warm-blooded nature, will help to determine whether this is a common trait or if it's a rare trait.
Thank you.
So this is great. We were sitting around late one night, downloading video, and a tech saw this. We were not expecting anything to happen with this. This is just going to show pictures of mud, basically. And it came in, and we had a great night then. So what does all this mean for larger studies in trenches? I think, as I've been saying more, we have really started to find evidence that shows that trenches are areas of high food deposition, that the steep nature are tending to funnel organic matter in there, which means that there's a potential for greater total biomass, even greater than in abyssal regions. We consider a lot of the deep sea to be food limited, to be deserts of food. But in these trenches, we might actually have processes of geological and physical nature that increase the number of animals. Hopefully, we'll be able to continue these studies and do more research. We also found out that megafauna rates of respiration are consistent with shallower counterparts. So that maybe a sea cucumber living at 2,000 meters may breathe just about the same amount of oxygen as a deep living one, assuming that they all get a similar amount of food. And so this means that there could be a large amount of carbon turnover by these resident trench fauna, which means that they could have a very important impact on how carbon is cycled across the ocean as a whole. And we know that now that the respiration by sediments indicates that these trenches sequester far greater amounts of carbon. And this, of course, is an potentially important avenue for sequestering carbon in the ocean. And while we can't say anything about whether this is going to mitigate larger carbon impacts at the surface or anything like that, it's definitely another area that we can add to our ever-growing budget and models of how carbon, oxygen, and other elements are cycled throughout the globe. So it's also important to note some of the perils. The implosion of HOV Nereus at 2,000 meters from the Kermadec Trench was very bad. We didn't get to do quite as much research as we wanted to, but it did spur us to create a new vehicle, the FVCR. Unfortunately, that was lost too in the Mariana Trench. And we have no idea where it is. We sent it down after a very successful experiment, and it never came back. So sometimes it's better to know what happened to your instrument in the deep sea, because, I mean, I've had sleepless nights wondering where the FVCR might be coming. But that's peril of any deep sea research. Of course, in these trenches, it is much more likely to happen because of extreme pressures. And the other parts of it, it's difficult to repeat this. Funding right now is not great for this type of research. We have had some very good work from the National Science Foundation and the Schmidt Oceanum Institute to provide research money for these two things. But it also means that we can't take as many samples as we want. There's always a potential loss of our equipment. And of course, that equipment is very costly to design. So before I end and take any more questions, I just want to thank all of our partners in this that have been very important to this research. Woods Hole Oceanographic Institute, Tim Schenk and Casey Machado helped us design this. Casey designed and piloted the HROV, and that was built by the Deep Submergence Laboratory. Most of these videos that you've seen of the baited traps with the snailfish, that is by Alan Jameson and Tom Lindley, the University of Aberdeen in Ocean Lab. Jeff McKenzie and Jeff Drazen and Mackenzie Geringer work at the University of Hawaii here at Manoa. And they're integral in this research. Like I said, Schmidt Ocean Institute, and the captain and crew of the RV Falcor, which is our research vehicle, ship, are very nice to provide this for us. The National Science Foundation and the University of Washington also provide a research vessel for us. So that's it. Thank you.
Do you have any questions?
Yes.
The Schmidt Ocean Institute, where is that and what is it composed of?
It's in California, and it's actually started by the Schmidt brothers. The older brothers started Google. So this Google group has their own research institution now. They're loosely based. They don't have like a main center that you can go see, like Monterey Bay or Scripps or Whistle or anything or things like that. But what they have done is privatize ocean research, which is a very good thing. Right? And there's definitely funding the government agencies isn't always reliable. There's definitely a question about whether that should be a primary function of the government. But so Schmidt Ocean Institute has outfitted the research vessel, and they are providing that for researchers all over the world. So it's a very good international group that's around this. Yes?
What is it about your physiology or body structure that allows you to withstand the pressure? And if you did catch one, it must be a trick to get it up to the surface. How long would that take?
Well, they never survive when they get to the surface. Yeah. So that's part of it. Part of it is just the releasing pressure. It's incredibly damaging to that because you're so used to having this 1,000 atmospheric pressures on it. So they hardly survive, but also the water temperature begins to warm up. So you're used to the temperature. So we think to some degree that they have biochemical adaptations, namely the amount of TMAO, but other osmolytes that allow their cellular functions to transfer pressure. Whereas if you took a fish from 500 meters to 4,000 meters and took it that deep, it wouldn't have the same concentrations of that material, so it would probably die.
Is there a depth at which they die versus
The theoretical limit of amount of this TMAO that you can incorporate into fish is right around maybe 200 meters. We've got very close to toxin fish. From 8150, we've seen this deepest living fish so slightly deeper than that. But beyond that, then their whole osmotic balance destroyed. So otherwise, it would be leaking fluid once you get deeper than that. You start incorporating that. So we think that to some degree, these sailfish, for some reason or other, they can just incorporate more of this chemical, and that allows them to be fish. Because all the other fish, ragtails and reindeers and other fish, they start to disappear around 6,500 to 7,000 meters in this almost exclusively sailfish. Yeah.
This probably will sound silly, but it's possible that with the lander, not with Nereus, but it might have been some big creature that wasn't on camera.
That's great. That's actually a great question. That's one of the theories of deep-sea biology to get gigantism. Of course, unless it shows up in the camera, we won't actually know about that. My theory is you are getting speaking about stuff. But you never know how this works. There have been lots of instances of animals causing major malfunctions in vehicles, especially in the oil and gas industry. They use deep ROVs for a lot of stuff. There are animals that seem like
How about the current down?
it's actually very slow. We do get to some areas where this trench, there's a funneling effect, but these trenches are actually so huge, it's hard to show that there. You could fit the Grand Canyon in these trenches. So they're not only very deep, they're very wide. So they're not like this narrow the crevice in the bottom of the ocean are actually very large.
So it showed, in the movie, it showed that there was some, when the fish went by, there a
Right.
little bit of muck that came up and just kind of,
Yeah, so that's probably on the order of maybe half a knot to a knot of current, which is not very much. That would move a particle 10 centimeters a second. Not too far, about this far per second. So some things might get washed away, but then there's always creatures like jellyfish and other things that are just never personally.
Yeah, Mariana is getting any deeper each year with the plates moving.
You think it is? So it's a subduction, like I said, the thing, they're actually dragging itself before these plates started to abut. They're probably very close to being at the maximum depth of the abyssal ocean, which is about 6,000 meters. And as that process increases, not only is it dragging itself down, it's pushing up the other side. So to some degree, that can actually push up the ocean mountains. But for the most part, what it does is cause volcanic eruptions that creates islands along these arcs. So yeah, so we know that some areas are getting deeper because of this geological processes.
the animals that are bigger depth become less complex. We saw fish, it's a pretty complex world.
No, the invertebrates are incredibly resilient. So that's what we see at the very bottom. And in fact, even the giant amphipod, he disappears at about 9,000 meters, and all you get are other amphipods and some shrimp. And that release from predation pressure makes it so that there's maybe 4 species there that are most common. Of course, we're still analyzing all the research and getting these things. And so what we see is that these amphipods, they're attracted to any type of food, whether it's a dead fish that falls all the way from the surface, or if it's just some sort of organic matter like plankton. We can use some of these things as opposed to islands. And so what we see that some of these amphipods, they exclusively feed on debris, and some of them are cannibalistic. I wish I could show lots of videos because there's all kinds of behavior. We've seen large shrimp go and pull amphipods off a piece of bait and swim off with it. We've seen other amphipods cannibalize each other. So I mean, it's actually a very competitive world down there, but that's because there's very little food. So we have to do lots of different things.
How would they be able to see each other or go and look at stuff if they can't see?
We think that chemosensory, so being able to smell the bait is very important. And a lot of ways that we estimate total populations is just by modeling this odor plume and saying, if this fish is this smelly, it's got this type of chemosensory response, we can determine how long it takes an animal to first reach the bait to see how it goes.
How's the water chemistry different from those surface?
It's not much different. The salinity is very similar. To some degree in these deep trenches, especially in the Southern Ocean, they have more oxygen because you have deep, dense water from the Antarctic, the Southern Ocean that flow into them. But otherwise, the chemistry doesn't change all that much. The one most notable cause is there's a very lack of calcium carbonate, which is important for forming shells. And that's because below a certain pressure in the ocean, it begins to dissolve. So it's very dependent on temperature and pressure. And so many animals have very thin shells in the very deep ocean below this carbon condensation. Yes?
What animals live in the mud? Do you find any, and do they go down to what depth do they go?
So we know that invertebrates live in the mud everywhere. There's lots of bacteria. There's a very small amount of these polychaete worms and some other things. But one thing that's ubiquitous both in the very deepest spots of the ocean to the very highest mountaintops are nematodes. Nematodes are roundworms. They're very simple. A great natural philosopher in the early 19th century that said if you remove everything from the earth, lakes, trees, grasses, oceans, and left only nematodes, you can still be able to see all the valleys, all the lakes, all the streams. So we know that those are everywhere. And in fact, in the trenches, they seem to be incredibly common.
And how much did that rover that exploded cost?
The Americans started out at about $1.5 million, and they got some donated cameras from James Cameron. created the gate going to the bottom of the Mariana Trench. So they're thinking the total loss was maybe about 3.5 years.
Over how long of a lifespan?
Yeah, it was around for maybe about 10 years, operationally 4 or so. So hard to say with things like that because we actually didn't have very good luck with the Kermadec Trench. It had about an 85% failure rate. And by that I mean get to the bottom of the trench and then the cable, the fiber optic cable, which is barely as big as a piece of bread and snap. So it was having some operational failures at that point in time. So it's hard to say what its actual overall effective use was. Some places it did very well in shallower depths, but I think the most important part of that is it did bring lots of whole new technology, which will hopefully be something that we can use in the future. So to some degree, the imagination used to come up with it as well.
Do you think with more time, you could make a version of the incubator that could actually bring the species back alive?
Yes, it can be very hard, and we have tried that with fish. Unfortunately, these things have to be built like tanks.
They have to be a closed system.
Yeah, so what they would do is they would, in fact, there's a pretty neat mechanism where you have a piece of bait on a hook, and a fish will come by and grab it and reel it into the chamber. And the doors will close and keep it at pressure until you get it back to the surface. But like I said, to be able to keep it at that intense pressure, the walls are several feet for this thing. Exactly. And the windows. Well, yeah, the windows, or you can put a camera inside there if you want to see. So that's one avenue that's always going to be important in deep sea biology.